Okay, so we are continuing on with our peppered moth example and how when the environment has changed, the evolution of the organism changes. So the most recent data says that 90% of the peppered moths are back to the non-melanic version or the lighter color of moth. So the gene frequency has changed and the frequencies and the calculations of those genes in the population do not match up with Hardy-Weinberg equilibrium. We can see mathematically that evolution has occurred. So let's move on to a human example of not only natural selection, but a mutation as well. So we've been talking about the sickle cell trait and how it's due to a mutation, and how malaria is the environmental factor leading to the success of that mutation, basically. So we're going to go through this whole story step by step. So first, let's look at this gene. There is a gene located on chromosome 11 that is known as hemoglobin S and codes for the production of the protein hemoglobin, which assists with the transport of oxygen around our bodies via red blood cells. However, millions of people have a mutated version of this gene and suffer from sickle cell anemia, which causes misshapen red blood cells, as shown in this picture. And this causes serious health consequences. A normal blood cell is round and moves smoothly through our veins and arteries. As you can imagine, the sickle cell shape causes problems. Okay. So what happens here is there was a mutation, and in this mutation, the normal version, there is a T, and in the sickle cell DNA, there is an A. This results in the transcription of a U instead of an A, and instead of the normal hemoglobin that we're seeing here, and the normal amino acid, so the normal amino acid is the glutamic acid, resulting in normal red blood cells. Valine is the amino acid produced, and it results in the sickle cell shape of the red blood cells. So it causes a change in the functioning of the hemoglobin so that it cannot bind to oxygen. So it doesn't just affect the shape, but it affects the way it binds to oxygen. So why would such a detrimental gene persist in the gene pool? Wouldn't natural selection have eliminated that gene? First, let's look at what causes it, and we did see that in the slide previously. There are hundreds of base pairs in the hemoglobin S gene, but all it takes is just 1 point mutation from a T to an A, which results in valine instead of glutamate in the resulting hemoglobin molecule. So we already talked about this, but we're looking at how a single point mutation changes the amino acid that is produced by this codon, and as a result changes the shape of the red blood cell. So this amino acid change results in a slightly different shape to the hemoglobin, which causes the red blood cells to be misshapen and causes them to clog up capillaries, cutting off oxygen to vital tissues. So let's look here at some key points. Those who produce normal hemoglobin have the genotype of either AA or AS. So heterozygotes have no serious complications even though they carry the sickle cell allele. So remember some of our vocabulary from the basics of genetics. AA would be homozygous, AS would be heterozygous because there are two different versions of the allele. So we're looking at the genes, we're looking at if they have the normal hemoglobin or the mutated version. AA would have 2 normal copies of this gene, AS would have 1 normal copy and 1 mutated version, the sickle cell version. So if you are an AA or an AS, you have no serious complications and you will have normal hemoglobin. However, if you have both recessive copies, the 2 mutated versions, you will have sickle cell anemia, which is fatal for 80% of untreated individuals before they reach reproductive age. So in a situation where you cannot get treatment, you will die before you reach reproductive age. There's an 80% chance that you will die, before you reach reproductive age. That is important to remember. So how are people surviving? How is this mutation still in the population? So that is the big question. Why hasn't natural selection eliminated the S allele, this mutation, from the population? So this has something to do with malaria. Malaria is also very dangerous and fatal, caused by the transmission of a parasite known as Plasmodium to humans through the bite of an infected mosquito. As shown in the next slide, when a human is bitten by a mosquito, the parasite can be introduced into the human host. So this diagram shows you how people become infected with malaria. So the mosquito bites a human, passing sporozoites to the new host. The sporozoite is a motile form of the parasite. The sporozoites travel through the bloodstream to the liver. In the liver, they create thousands of merozoites, which are the daughter cells of that parasite. Then the merozoites enter the bloodstream and infect the red blood cells. Within these cells, the merozoites continue to multiply, eventually causing the cells to rupture. The merozoites then invade other red blood cells in the bloodstream, and the cycle continues. Over time, some of the merozoites develop into male and female gametocytes, which may be transferred to other mosquitoes that bite the human, and those can go and produce gametes or sex cells, and within the mosquito, sexual reproduction occurs, and then the whole process starts over again. The newly formed sporozoites, I don't know why that's so hard to say, travel to the salivary glands of the mosquito where they can be transmitted to the next human. So this is kind of the life cycle of the parasite that causes malaria. Okay, so I said all of this basically, and this is how malaria is spread, and it is extremely dangerous and kills close to 1 million people every year. So we are looking at two different maps. One showing that S allele, don't forget that's the sickle cell mutation, and that's in the colorful map of Africa here. The other map is showing areas where malaria is present, and you can see that these areas match up almost identically. So going back to a simple review of what we're talking about, homozygous means you have two copies of the same allele of a gene. Heterozygous means you have different alleles for the same gene. So here we're seeing two homozygous versions and one heterozygous. Homozygous dominant would be the AA. So two normal copies of the hemoglobin gene with no mutation. You have normal red blood cells. With SS, you have two copies of the recessive gene, which is the mutated gene that causes sickle cell anemia. AS would be the heterozygous version of this gene, which means you have different alleles for that gene. And as you remember when we were talking about the sickle cell trait or sickle cell mutation, if you have one normal copy and one mutated copy, you are not afflicted with sickle cell anemia. So let's continue here. The heterozygous dominant or the homozygous dominant normal hemoglobin individuals are highly susceptible to dying from malaria. Individuals homozygous for sickle cell anemia do not survive to reproduce. However, individuals that are heterozygous for normal hemoglobin and sickle cell mutation either did not contract malaria or suffered a less severe malarial infection. So what we're finding out is that if you have the sickle cell trait, not the disease, it provides some protection against malaria because of the way malaria spreads and uses the blood cells as part of its life cycle. This sickle cell trait is providing an adaptive advantage. So let's think back to our map. We're seeing that sickle cell trait and malaria have some sort of connection. And let's go back to the story here. Beginning in the 1940s and 1950s, researchers began to notice that humans with the faulty S version of the hemoglobin gene tended to live in equatorial Africa, where malaria is also common. Not only that, but people with the heterozygous version, AS, also tended to be most frequent in those areas. What happens is those individuals tend to not die from malaria as often as individuals who produce normal red blood cells. It was a young researcher by the name of Anthony Allison who figured this all out. He figured out that individuals with normal red blood cells were susceptible to malaria. Individuals who were homozygous for the faulty hemoglobin gene often died of sickle cell anemia. But the heterozygous individuals could survive both. It turns out that the AS individuals produce red blood cells that do not sickle, but are poor hosts for malaria. For some reason, the AS cells are poor hosts for the parasite that causes malaria. And this results in what is known as a balanced polymorphism, in which the heterozygous individuals are the most fit. So the balance here is that both traits are persistent, both the normal hemoglobin and the altered version that causes sickle cell anemia. Therefore, both traits persist in the population because the heterozygous genotype is more common. So if two surviving heterozygous parents have a child, there's a 25% that the child will inherit the S allele from the mother and the S allele from the father and have sickle cell anemia. There's also a 25% chance that they will inherit the A allele from the mother and the A allele from the father, leaving them susceptible to malaria. But both of those alleles remain in the population since the heterozygous individuals have the highest fitness. So, there are other examples and other adaptations to malaria. Since malaria is such a detrimental and dangerous environmental factor, there have been other mutations that protect against malaria as well. So here on the map, we're seeing some other versions, other mutations to protect or adapt against malaria. Okay, we are moving on. That was natural selection. We're moving on to another function of evolution or force of evolution called genetic drift. So, a real quick definition before we move on to the next part of the lesson. Genetic drift is a change in gene frequency in a population caused by a random event. That is important to remember. The traits don't matter, the environmental factors don't matter, randomness is what matters. So in this picture, it doesn't matter if beetles are green or beetles are orange or brown. They got smashed randomly because of a random event. And you can see in the final circle here, the proportion, the gene frequency in this population would have changed due to the random event. So this concludes this part of the lesson. I think we are moving on to part 5. So please proceed on to the next part of this lesson on forces of evolution.